This is also relatively unique in that it's very multi-jurisdictional. We have state interests, tribal interests, federal interests, provincial interests, local and municipal interests. So fish are always moving across different management jurisdictions, and we have to account for that in our decision making, but that's very challenging in ecosystems like the Great Lakes. So the way we've typically gone about measuring fish movement over the years is through the use of some kind of external mark. These are pictures of a walleye. Walleyes are kind of pin cushions. Anything that can be possibly attached or pinned to them have been. These are examples of two different type of tags or marks that have been used in the past. The top one is a jaw tag, which is just a metal clamp that goes around the jaw of the fish. And then the bottom picture shows probably something you've seen before, which is a floy tag or a spaghetti tag that gets pressed into the musculature and skeleton of the fin and contains an ID number which can be used to identify that individual. Now, one of the challenges in talking about a technology like acoustic telemetry is not a magic bullet. It overcomes some problems for sure. And this form of marking fish is still perfectly fine. It answers a lot of questions that we need answered. In particular, over on our side of the pond, there's a big stocking program, for example, for Atlantic salmon, and they're mostly concerned with tracking where angler captures of these stocked Atlantic salmon come from, and this works just fine for that purpose. But a limitation of this particular way of doing things is that it really only gives you two different information points. You know a location and a time where the fish was marked and released, and then you let it go, and at some other time, at time T, whatever that be days or years from the release, it's recaptured at point B. Now, that's all well and good, but what it doesn't give you is, let's see, I'm right-handed, but I'm going to force myself to use the pointer left side here. What it doesn't tell you is anything the fish does between A and B. And secondly, in many cases when that fish is caught, often it's by an angler, a recreational or commercial angler, it's then removed from the population. So the data you're forced to work with is just two individual points. And while that works for some analysis, that detection history of just 2 points in time isn't the most informative and doesn't lend to very robust analysis of population parameters like survival and migration rates and that kind of thing. It also doesn't give us any information about where the animal spent its time, which could be important in the case of habitat protection or restoration. So that's where the limitation of this way of doing things lies. So acoustic telemetry is a way, basically addresses those particular problems. In acoustic telemetry, fish are captured. They are, just like with the external tags, they're marked with a tag, but in this case it's an electronic tag. And in most cases, this tag is surgically implanted into the body cavity of the fish. There are some animals where you can actually attach a tag like this externally, but that runs into problems when it gets growth or biofouling on it and that kind of. This tends to be the better way to do this, and as long as you're using trained surgeons and good sterile techniques, the fish recover and heal very well from this process. These acoustic tags come in all shapes and sizes. That picture there is a adult lake sturgeon, which is getting a tag that looks like that. It's just a little bit longer and a little bit skinnier than a roll of lifesavers. It's a very powerful tag. You can hear this thing, depending on circumstances, in some cases for over 1.5 kilometers away. So it's a very powerful, strong tag. that's something that might go into an adult lake sturgeon or adult lake trout. They scale all the way down to something like this, where maybe this is a smaller walleye, smallmouth bass. Then you can take another step down, and maybe you're, I don't know, yellow perch or something like that, white bass, and then the smallest one, at least from this particular manufacturer, you probably can't even see this, but it's basically a centimeter long, and it's basically looks like a squished Tic Tac. So very, very tiny. So you can do this with a wide variety of fish sizes, and as the battery and technology advances, we may even be able to get smaller than this. The trade-off is that this little guy only lasts for like 50 days tops, whereas you can get, depending on how you program, you can get up over 10 years on a tag like that. So lots of options, which creates a lot of choices for researchers, but it's pretty flexible technology in terms of the species size of fish you can use this with. So after you've got your fish tagged, you let it go. And then in most applications of this technology, the fish then is detected on these stationary networks of acoustic receivers. Doing things this way is called passive acoustic telemetry, because the fish moves past stationary hydrophones or receivers. There's something else called active or manual, where you can actually take one of these hydrophones and go in a boat and track fish around. That tends to be more labor intensive, and so most people go this particular route. So every time a fish swims past one of these receivers, each one of these tags has an individual code. So you not only know that that a tagged fish was somewhere in the detection radius of these receivers, but you also know that this was Fred or Betty or Ethel, the lake sturgeon. Each tag has a unique code attached to it. Now, it should be pretty obvious that in relation to that external tagging, Mark, what you get with the acoustic telemetry is with an regular external tag, you'd know, say you released it here, and then you caught it in this tributary up here, you'd have a capture observation point there and one here. But with the acoustic telemetry, obviously you get then more of a pathway. So this technology tends to be very powerful for addressing management problems and questions where the route or the pathway of the fish is something that's important. This got some of the highest profile science that this technology has been used in was out west, where salmon smolts had to migrate downstream through turbines, and basically their choice of turning left or right was the difference between living and becoming chowder. So obviously that's a case where this path selection by the fish is very important, and acoustic telemetry is very powerful in those kinds of powerful tool in those kinds of scenarios. So of course then once you've gathered some information on where these fish are and when, you can start asking some basic questions about where are the fish, when do they move, how do they move, why do they move. So all the data you get is really just a series of timestamps attached to a particular location, which then you construct movement histories with. Some of the tags that are now being manufactured, you can also add a pressure sensor, so you can tell whenever a fish passes by one of those receivers, the tag will not only report the ID code, but also the depth of the animal. So if you pack receivers really close together, so you have overlapping detection, you can actually get 3D paths of fish when you analyze it. So this is kind of a mock track of a tagged sea lamprey migrating through a channel of the St. Mary's River. You can add temperature sensors, acceleration sensors if you're interested about activity and swimming speed. And the latest wrinkle in these things is a new kind of tag called a predation tag, which contains a dissolvable like burn wire. And when you put a predation tag into a small fish, say a cisco, and it gets eaten by a lake trout, that burn wire dissolves in the stomach acid of the lake trout. And once that burn wire gets eaten through, the signal that the tag emits will change from I've not been eaten to I've been eaten. So it is obviously a numeric code, right? But so these are brand new. There's not very many studies that have used them, but they're unique in the sense that they start to get you at species rather than just asking where fish go. You can now do predation studies, which is a particular type of species interaction, which is pretty exciting. So some of the popular kind of theme areas that telemetry studies get organized around to answer are things related to habitat use, activity, response to disturbance. These are all basically questions that relate to behavioral ecology. But when you do take the data and do more modeling with it, you can also get at start calculating basic population dynamics parameters like mortality, migration rates, and site fidelity. And then once you have that information, then you can begin applying that to projects that are designed to restore, manage, and deal with particular threats. So I'll try to give you some specific examples of how that works here in a moment. So acoustic telemetry was not used in the Great Lakes very much prior to 2010, maybe 2 to 3 studies. It's a tool used very often and commonly by fishery biologists in marine environments, and there was some of these early networks which were largely focused on the salmon issue out west, the California Fish Tracking Consortium, the Pacific Ocean Shelf Tracking Project, and the Ocean Tracking Network. So these were collaborations of researchers that were using acoustic telemetry to address fishery management and conservation issues in marine environments. But none of that technology had migrated to the Great Lakes, or that expertise had migrated to the Great Lakes prior to 2010. What changed that was the advent of the passage of this program here, the Great Lakes Restoration Initiative, which I'm sure many of you have heard of. This was a Obama-era legislation that had a number of objectives, but at the ground level, what it did was pump millions of dollars into aquatic and ecological restoration science in the Great Lakes. This was money at the scale that hadn't previously been seen before. And fortunately, at the time, there were some very good minds at the Great Lakes Fish Commission who are aware of the acoustic telemetry programs going on in marine environments, and thought that with the advent of GLRI, that now was the time to see if we could begin importing this technology and this expertise into the Great Lakes. So they put together some large proposals. They were successful at getting GLRI funding, and they basically laid the groundwork for this organization that you'll hear me call GLADOS, which stands for the Great Lakes Acoustic Telemetry Observation System. The way it all began was with 4 core projects. There was a sea lamprey project that was using acoustic tagged sea lamprey in the St. Mary's River to begin understanding how to better control this invasive species in that very large river system. A second project looked at lake trout spawning behavior in the Drummond Island region of northern Lake Huron, specifically looking at whether there was differences in the behavior of naturally produced and stocked lake sturgeon to try to help better inform stocking strategy. A third project looked at walleye source dynamics between Lake Huron and Lake Erie. And then the last project, which is a project that I oversaw, was one that looked at lake sturgeon population structure in the Detroit-St. Clair River system. So these projects got started with some fairly substantial funding, and through meetings and a lot of effort on the part of the Great Lakes Commission, our Great Lakes Fishery Commission, we were able to convince folks that this was perhaps a technology that would apply not only to these particular projects, but also something that could be benefit elsewhere in the Great Lakes. The mission of GLADOS was to first conduct novel research, and by novel we mean large scale, at scales not previously, that we previously weren't able to conduct movement research in the past. But then secondly was to establish a network of researchers to do three different things. First, to develop a collaboration or a community of collaborative partnerships to develop and increase regional telemetry experience, so that there was not just a few experts, but many people who were wise and capable in deploying this technology in different contexts, and also to facilitate data sharing amongst projects. And I want to kind of just focus on this data sharing aspect and for a moment, because this is one of the powers and great things about GLADOS, and this can all be kind of summed up as tagging locally but observing globally. So let me give you a little example of what I'm talking about here. So this is Tom, my good friend and colleague Tom Binder. He's happily tagging his lake trout up in the Drummond Island Refuge. He's got his receivers. He's got his receivers in the area he's interested in all set up, and he's happily going along, doing his work on his project. Well, other people hear about the really interesting work that Tom is doing, and they start developing acoustic telemetry projects all over the basin. You can think of each one of these stars as perhaps a location where receivers or a small acoustic array might be located. So the thing is that Tom's fish, Tom is getting data that he needs and he desires from the area that he's most interested in, but his fish most likely, or could be, meandering around these other projects and registering detections on other people's receivers. And so you can see how this quickly can turn into a giant mess when you have a number of projects all ongoing. You've got detections of your fish on your receivers, but now you've also got, as other people develop projects, now you've got detections from other people's fish on your receivers and your detections of your fish on other folks' receivers. And how do you keep this all straight and organized? And that's one of the things, one of the major functions that GLADOS performs. So all of us who are part of GLADOS then take all of our data, ship it off to GLADOS Central. All that information gets run through very large scripts and databases, and then after a while, some kind of papal smoke floats out of a window, and then you get a data file back from GLADOS that contains not only the detections of your fish on your receivers, but also detections of your fish on anybody else's receivers anywhere else in the network. And there's still groups out. I was actually a couple of years ago at a meeting in the Mid-Atlantic region where they were still trying to do this process by literally shuttling spreadsheets of information back and forth between hundreds of different researchers, and you can imagine how that could get really confusing and messy and lead to errors. So So this collaboration, the being willing to buy into data sharing is really key to implementing this technology in a really wise fashion. So that's the data file going back to Tom. Okay, so from humble beginnings, 4 projects within the GLaDOS portfolio. There are now over 70 projects. There's 269 scientists who are actively participating data. Those scientists collectively represent 69 different organizations. So there's been amazing amount of growth and buy-in. Just to give you a more visual illustration as to how GLaDOS has grown over the years, we give you a little animation show you a little animation here where the blue dot, each blue dot is a receiver deployment. Keep an eye on Lake Erie, which has the most dramatic, but can basically see the Great Lakes kind of filling up here with receivers. For a time here, there was a little line right offshore where we are. There goes Lake Erie filling up with receivers. So you see the potential for doing the kinds of lake-wide movement studies that weren't previously possible without this technology. So just a few other quick bean counting things. So far within GLaDOS, there's been 44 different species tagged. The top 4 are walleye, sea lamprey, lake sturgeon, and lake trout, and there's a whole host of other ones. A grand total of over 11,000 or nearly 12,000 fish have been tagged and released. There's all different ways of calculating the number of receiver deployments, which you probably don't care about, but then the centralized database now contains over 280 million detections from individual fish, which is just an astronomically large data set. And most of these projects generate data in abundance far greater than anybody expected, and that actually has become a real analysis challenge about how to manage and analyze all this information. We've been, I think, productive scientifically. We've put together over 50 peer-reviewed publications. Hopefully the curve isn't tapering out. I was looking at this last night. I was thinking, okay, well, here's the initiation, then people get excited and like, whoa, now this is a lot of work. Now it's starting to taper off, right? But hopefully that curve still trends upwards. But not only are we doing this work, but we're getting the messaging, we're getting the findings out into the scientific literature where people can use it. All right, so just to round things out, I want to provide you just a few more. It's great to produce papers. Most of you aren't in the scientific, you're not nerds like me. You're not going home on a Friday night and reading, grabbing your issue of the Canadian Journal of Fisheries and Aquatic Sciences. You should, but you're probably not. So let me give you some real practical examples of how this information is beginning to affect management decisions. This first one comes from Lake Erie. Walleye there are the bread and butter. They support a commercial and recreational fishery that's valued in the billions with a B. So very, very lucrative fishery, very important to the economy. This is a map of Lake Erie showing the different management units that the lake managers on both sides of the border use to manage these fisheries. And the thing to note here is that, I don't know if you can read it from the back, but MU 1, 2, and 3 are managed with a quota system. So folks fishing in those particular zones, each year they're issued a particular quota that they're not to exceed, their catch is not to exceed. So that we call that the MU through 1, 2, and 3 are the harvest management zone, And for a long time, the amount of fish that were allocated to those particular zones was kind of, it was tacitly made under the assumption that fish weren't leaving those zones into the eastern basin. Now, these guys aren't stupid. They knew that, you know, this did happen to some degree, but they weren't really sure. They thought it was not significant enough to really worry about. And there was also the assumption that if that fish allocated to Unit 2, for example, are primarily harvestable in Unit 2, and they aren't, you know, skedaddling off to Unit 1 or Unit 3. So when they actually began to tag these fish and get a sense for their movements, they found, you know, something very different. So from left to this, the quota zone is basically to the left of this, so all this in here. So you see a lot of these fish moving off into the eastern basin. That was a little bit higher than they expected, but not to a huge degree. The concern actually was here is that a lot of these fish within these first 3 management units were actually not harvestable in some units during time, at one point of the year, but harvestable in others. So it really opened their eyes that, you know, just because we issued a quota to a particular unit doesn't necessarily mean that those fish are actually there all the time to be harvested. And that was something that really needed to get worked into the harvest management plan, and they've recently adjusted quotas based on some of the information that they've gathered from this large-scale telemetry study. So a little closer to home, Lake Huron, there's been a couple of lake trout projects, fairly high-profile lake sturgeon or lake trout projects. One focused on the spawning population in the Drummond Island Reserve, and then another one in the Thunder Bay area. The project in the Drummond Island area was focused on understanding the reproductive behavior of stocked and wild lake trout, and also the role that the reserve played in protecting these fish from commercial and recreational harvest. And then the Thunder Bay project was focused on understanding, you know, how could we recreate spawning habitat that was lost? What did that need to look like? How should we construct it? In the Drummond Island project, this is a map showing the lake bottom bathymetry. The cooler colors are deeper. The orange, yellows, and reds are shallower water. If you'll note that these like finger-like projections of shallow water coming out from the shore, these are geologic formations called drumlins. I feel like, you know, I'm from geological survey, so you got to point something geologically out, right? So they had a fine scale array where they could really locate and locate each tagged lake trout at a very small, fine scale in this network. And when they looked at where all these fish were located, they were actually able to determine where these fish were actually spawning within this reserve. So obviously you can see that not all spawning habitat in the reserve is created equal. These fish are zeroing in on certain areas of it, and they're actually a fairly small percentage of the overall habitat. So then they were able to take that information and then get a sense of what these fish are actually looking for when they're spawning, and determine whether wild fish and stocked fish were using this habitat in the same way. Moving down to Thunder Bay, this is more bathymetry again. These shallower areas here that say degraded, these are former lake trout spawning habitats that were ruined by dumping of the concrete plant over here that dumped, I don't exactly know what they dumped, but they dumped something that filled in the crevices and ruined that habitat. So there's a large research consortium of folks who got funding together to build different types of reconstructed spawning habitat, but they weren't really sure exactly what was going to attract or concentrate these fish. So they made all of these made an array of test spawning locations with different designs, and then used acoustic telemetry to monitor lake trout use, or let the lake trout determine, which of these areas they preferred. And don't worry about all the text and the bars here. Basically, this is just the different sites and showing the numbers or the proportion of tag fish that we're using each site in different years. The point is by being able to use acoustic telemetry to monitor those different experimental sites, they were actually able to get a sense in quantitative terms about what a lake trout spawning site, what characteristics it should have if you wanted to replicate this in the other areas of the lake, which would be very useful information. One more example, this one on invasive species control. I'm sure you're all familiar with lamprey. Scott just reminded us it was one of the major players in the decline of, lake trout in the Great Lakes. They're parasitic. They do really bad things to large-bodied fish. The St. Mary's River is one of the largest lamprey producers in the Lake Huron system, but most of it's too big to treat with chemical lampricides. So they were intensively trapping lamprey in here to remove them from the system. A project led by Chris Holbrook was able to use acoustic telemetry to show which pathways most lamprey were taking, so that you could more effectively target choke points where you could get the highest catch of lamprey and remove them. So this is another case of the spatial pathways being key information here. And then lastly, a project that I led on the Detroit and St. Clair River. The system is habitat for a large population of lake sturgeon. They're genetically differential. They are not genetically differentiable, but there's been some question as to whether there are single or multiple different populations present in this system. And when we looked at their movements, what we found was that although they're genetically similar, fish tagged in different parts of this system still sort of behave as different populations. So these black dots are fish caught and released in the Detroit River, and they don't seem to interact very much with the red dots, which are fish captured and released into the lower St. Clair River. And then these fish and the blue dots, which is probably hard to see with the lights on, are upper St. Clair River fish, and they're the most wide-ranging. Some of these guys actually did get detected on that Bruce Peninsula to Manitoulin Island array that was in place for a while. So while these fish are genetically similar, they don't behave that way. They still behave as spatially structured groups of individuals. And there we go. Okay, so if you're curious about this, I can give you some more information. There's also been work done. A recent liberalization in walleye harvest in Saginaw Bay was a direct result of acoustic telemetry studies. Acoustic telemetry is now being used on round goby and grass carp to better understand the invasion biology of these species, and it's also being used to try to get a better handle on the appropriate strategy and site selection for stocking of lake herring or cisco. So the future of GLaDOS, as I mentioned, Chuck Kruger, who is the current director, is stepping down, sailing off into retirement. Dr. Chris Vandergoot will be the new director. Both are super smart individuals. We'll certainly be in good hands in Chris's leadership. A challenge for us moving forward is that we're still heavily dependent on Great Lakes Restoration Initiative funding. Initially, the Trump administration proposed to to drastically reduce that. There was a ton of pushback. He acquiesced, and it's safe at the moment, but one never knows. So we probably need to find a way, and we've had discussions about this, to try to become part of somebody's base funding, but that's kind of an ongoing discussion, and we'll see how things happen. So thank you for your attention. There's the GLADOS website if you want more information, and I'm probably out of time, but questions after. Okay, thank you very much.